Good afternoon, students. Welcome to the online class of Biology for Engineers. In our last session, we have started with Unit 3, Genetics and Evolution. We shall continue with this Unit 3, Genetics and Evolution. The next topic, what we are going to study in this Unit 3 is cell division. Cell division, it is nothing but the division process where the cell is dividing into two progeny cells or the daughter cells. Cells will duplicate or show the process of division, and it will finally give rise to two daughter cells by this two process, that is mitosis and meiosis. So this process will mainly help in making new cells for growth and development of all the living organisms. Mitosis process of cell division, it will occur in the vegetative cells, and it will help in growth and repair of the tissues of the bodies of living organisms. As we know, all the living organisms are made up of cells, and group of cells will join together and form the tissues of the living organisms. Group of tissues will form the different organs. Different organs will combine together and form the complete living organisms. So the tissue of the bodies will be repaired by this process of mitosis, and mitosis will also help in the growth of the living organisms. Meiosis will occur in the reproductive cells. Meiosis process will occur in the germ cells, and it will help in reproduction process. So meiosis and mitosis are the two types of process of cell division. So mitosis will produce the daughter cells. Cells will divide by this mitosis process, and it will produce the two daughter cells, which the two daughter cells will be identical to the parent cells. And this meiosis will produce the two, not the two daughter cells. In the meiosis process, it will produce four daughter cells, and the daughter cells will not be identical to the parent cells. And this will help in growth and repair of the tissues of the organisms, living organisms, and meiosis will help in production of gametes, reproduction, and it will form the four daughter cells. Meiosis process of cell division. Cell cycle we have in cell division. Cell cycle is divided into two phases here. The first one is interphase, and the second one is mitotic phase. Mitotic phase we also call it as nuclear division phase. Here the nuclear division you can see in mitosis. Interphase is the phase in the cell cycle. Generally in the interphase, the cell will produce the different proteins and help in the further division of the cell cycle. Next we have this is the cell cycle, representation of the cell cycle, diagrammatic representation of cell cycle. This G1, G2 and M are the phases related to interphase. Interphase will have this G1, G2 and S phase. G1 here is gap phase 1 and S is synthesis phase. S means synthesis and G2 is gap 2 phase. Second growth phase, this is second growth phase G2 and G1 is first growth phase. G1 stands for gap phase 1 and it is the first growth phase. This is the interval between the mitosis and DNA replication. Here in G1 phase, so we were discussing about the cell cycle. This is the cell cycle. In the G1 phase of cell cycle, here you can say RNA and protein synthesis will takes place in the G1 phase. And G1 phase is followed by S phase, synthesis phase, where DNA synthesis is taking place in the S phase. That's why we are calling it as synthesis. DNA synthesis. Production of the DNA is taking place in the S phase of the cell cycle. Then we have after G1, S and G2 we have here gap 2, which is the second growth phase. In this RNA and protein synthesis, further RNA and protein synthesis will be taking place. The cells will be synthesizing the proteins which will be required for the mitosis and growth in size. In this G2 phase, proteins will be synthesized which will be required for the mitotic phase further, that is M here is mitotic phase. Mitotic phase again is having this 4 phases, that is mitotic phase is divided into 4 phases here. First one telophase, second one anaphase, third one metaphase, and fourth one is prophase. So four major phases you can see in mitosis. M. So this is mitosis. Mitotic phase. In this we have interphase. Interphase is having three phases. Just now we have seen G1 phase, S phase and G2 phase. In the interphase, which is the longest part of the cell cycle, and it is having the three phases, that is G1 phase. G1 here is gap 1 or the first growth phase. S synthesis phase where the DNA synthesis is taking place, and G2 gap 2 phase or second growth phase where the proteins are produced or synthesized, which will be required for this mitotic phase and the growth in size. Mitotic phase, you can see here, it is divided into karyokinesis and cytokinesis. Karyokinesis means the division of the nucleus. We have seen the cell. Inside the cell we have nucleus. In our previous session we have talked about the cell. So division of the nucleus is nothing but the karyokinesis, where the nucleus is dividing. Mitotic phase involving the division of nucleus, we call it as karyokinesis, and this cytokinesis is the division of cytoplasm. Cytokinesis is the division of cytoplasm. Cytoplasm is the fluid content of the cell. Division of cytoplasm, we are calling it as cytokinesis. So mitotic phase will give rise to two daughter cells. By this, four phases, that is prophase, metaphase, anaphase and telophase. This is about the stages of mitosis. In the mitosis we have discussed, it is having four stages, that is first one prophase, metaphase, anaphase and telophase. So we'll discuss about these stages of mitosis. This is the first one, prophase. So you can see here A, B and C involves, A to C is prophase. Prophase is the first phase of the karyokinesis. Pro means before. So you can see here this nucleus. Inside the nucleus, cell will have the nucleus, and inside the nucleus these are the chromosomes, which are thread-like structures. These chromosomes are thread-like structures in the nucleus. Then it is becoming, here you can see it is becoming somewhat thick. Chromosomes are becoming thick here. CHR here is the chromosomes which are present inside the nucleus, which is helping in cell division here. chromosomes. These are becoming thick. Chromosomes are becoming thick. This is CN is the centromere. Centromere. Chromosomes at the center will have the centromere, which will divide the chromosome into two arms. This chromosome will be dividing the chromosome into two arms. These two arms will be joined together by this centromeres. Next, this is D is metaphase. Meta means between. Here you can see SP, spindle fibers. Spindle fibers will be formed in the metaphase. Chromosomes here you can see with two chromatids joined by the centromere. The spindle fibers will be formed by the help of these chromosomes. They are aligning towards the equatorial phase of the cell. This arrangement of the chromosomes at the center we are seeing here in metaphase. This is the spindle fibers SP, and all the chromosomes are arranged here at the center in the metaphase, and this we are calling it as metaphase plate. These are the chromosomes and they are attaching at the center, and this attachment at the center we are calling it as equatorial plate. So we were discussing about the metaphase of the cell cycle, mitotic phase, then anaphase E. Here after metaphase we have E anaphase. Ana is moving up. You can see chromosomes in this, they are at the center, and in the anaphase E they are moving up. Ana means up, anaphase. The splitting of the sister chromatids of the chromosomes you can see here in the anaphase, and they are moving away from the equator. Center we are calling it as equator. If they are moving away from the center is the equator. Here they are moving away from the center, that is away from the equatorial plate, and they are moving at the ends. Ends we call it as poles. That means they are moving towards the poles. The chromosomes, migration of the daughter chromosomes towards the poles will occur in this anaphase. You can see these chromosomes are started to moving away from the center. Center we call it as equator, and this corners or the edges we call it as poles. You can see here they have moved near the poles. The chromosomes are moving near the poles. Then CP here is the cell plate which is formed finally and forming the two daughter cells. From one cell, two daughter cells are formed at the end of the mitosis. This is the telophase. Telo means end. This is the telo is the last phase of the mitosis. In the telophase, the new nuclear membrane will be surrounding the daughter cells, and the spindle fiber will finally disappear. These fibers will finally disappear in the telophase stage, which is the end stage of the mitosis, and finally divide the cell into two daughter cells. So this is about the stages of mitosis. A, B, C is the prophase, D is the metaphase, E is the anaphase, and the last one is the telophase. 4 stages of the mitosis. This mitosis process is very helpful in the growth of the organisms. It will repair all the body cells with the new cells. The RBC cells will be formed with the help of this mitosis process. Growth of the plant will be mainly, the continuous growth of the plant will be due to the mitotic phase that will occur in the tissues of the plants, or the cells of the plants will undergo this process of mitosis and form the new daughter cells. Next we have is the meiosis. Meiosis process, it is also very useful for production of gametes which will help in reproduction. Meiosis, you can see here, we have meiosis 1 and meiosis 2. In meiosis, 4 daughter cells will be produced. That's why it will be divided into meiosis 1 and meiosis 2. You can see here meiosis 1, first meiotic division. We also call it as heterotypic division or reduction division. Meiosis 2 is the second meiotic division or the reduction division. Meiosis 1 is divided into karyokinesis and cytokinesis. Just now we have discussed about karyokinesis is involving the division of the nucleus. Cytokinesis involves the division of cytoplasm. The fluid content of the cell is the cytoplasm. Next, in the karyokinesis, we have 4 phases. We have discussed just now. In mitosis also same like this. In meiosis we have 4 phases, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. Again in prophase 1 it is the longest and it is further divided into the following 5 substages. This substages division of the prophase 1: leptotene, zygotene, pacatene, diplotene and diakinesis. These are the substages, 5 substages of the prophase 1 of meiosis 1. Next we have meiosis 2. In the meiosis 2 also we have karyokinesis and cytokinesis. kinesis. Karyokinesis 2 is the division of the nucleus, and cytokinesis is the division of the cytoplasm. In the karyokinesis 2 also we have prophase 2, metaphase 2, anaphase 2 and telophase 2. As 4 daughter cells will be formed at the end of meiosis, that's why you can see here meiosis 1 and meiosis 2, resulting in the formation of 4 daughter cells. As in the case of mitosis, just now we have seen only 2 daughter cells we have seen in mitosis. So this is about meiosis. This is the process of meiosis 1 and meiosis 2. So in the prophase 1, we have the first substage, that is leptotene here. Leptotene, the first substage of the prophase 1 of meiosis 1. This is the cell. In the cell we have nucleus. Nucleus will be surrounded by a nuclear membrane. In the nucleus we have another structure called nucleolus, and in the nucleus we have chromosomes, which will mainly help in the division here. Chromosomes are thin, long, thread-like fibers in the leptotene stage, you can see. Then chromosomes are becoming thick in this zygotene stage of meiosis 1. Chromosomes is exhibiting synapses here. Synapses means pairing. Pairing of chromosomes is taking place in the zygotene stage of the meiosis 1. Then after pairing, it is forming tetrad, means four chromatids. A pair of chromosomes will consist of 4 chromatids. We call it as tetrad, because in a bivalent, chromatids of the same chromosome will be joined by this centromere. Show you the tetrad. This tetrad is formation of 4 chromatids. You can see here in this slide. This one, tetrad. This is the centromere of the chromosome, which will be helping in joining of the chromatids. These are the 4 chromatids of the chromosomes. This is bivalent. The 2 chromatids will be joined at the centromere. This is the centromere. Chromatids of the same chromosome, we call it as sister chromatids, and chromatids of different chromosomes, we call it as non-sister chromatids. chromatids of the same chromosome are sister chromatids, and chromatids of the different chromosomes. These are the chromatids, four chromatids of the chromosomes which are joined at the point called centromere. This is the crossing over between non-sister chromatids which is taking place the places at which the crossing over is taking place, we are calling it as chiasmata or the chiasma. This help in this crossing or exchanging will help in the exchange of the genetic material that will result in the production of the new recombinant chromosomes, or the genetic recombination will be taking place. Exchange of the genetic material will be resulting here by this chiasma, exchanging of the genetic material between the chromosomes. So this is the chiasmata formation between non-sister chromatids of the two homologous chromosomes. These are the chromosomes with four chromatids. We call it as tetrad. Chiasma is the exchange of the genetic material between the chromosomes. So we were discussing about meiosis. So these are the tetrads or the chromatids of the chromosome. Just now we have discussed. Then in the diplotin, the nuclear membrane and the nucleoli will begin to disappear here. Disappearing of the nucleolus, crossing over is taking place between non-sister chromatids. Just now I have shown the chiasma formation which is helping in exchange of the genetic material. Then we have the diakinesis, the substage of the prophase 1 of the meiosis 1. Then we have metaphase. In the metaphase, we have seen the arrangement of the chromosome at the center. Center we call it as what? Equatorial plate of the cell. The chromosome here, these are the chromosomes, they are attaching at the equatorial plate in the metaphase. This spindle formation will take place. Centromere is the joining of the chromatids at a point. This point will help in the formation of the tetrads. Chromosomes are aligning at the equator at the center in the metaphase. Chromosomes are arranged on the equator in the metaphase 1. Then in the anaphase, the spindle fiber will undergo contraction and the chromosomes will start moving away from the center and they will move up, up. They are moving towards the ends in the anaphase. Then we have the telophase. Telo is the end of the meiosis 1. That is telophase 1, where in the telophase 1 you can see at the end of the telophase the two daughter cells are forming here. So first meiotic division is resulting into formation of two daughter cells here, you can see. Then here, from here, meiosis 2 is starting with prophase 2. In the prophase 2 again, you can see the chromosomes, these chromosomes. This is the cell nucleus. Inside the nucleus we have chromosomes with chromatids, nucleolus, and nuclear membrane will start to disappear in the prophase 1. Then in the metaphase, again you can see in the metaphase the chromosomes are aligning at the equator. Chromosomes will align at the equator. They will arrange themselves at the equator. Then in anaphase they will start moving towards the edges at the ends due to contraction in the chromosomes. In the spindle fibers, the chromosomes will separate here and they are moving away from the equator in the anaphase. This is the continuation of meiosis 2. This is the telophase 2, telo end of the meiosis 2, where the daughter cells are formed here. The nuclear envelope and the nucleolus will reappear, and you can see here four daughter cells. Four daughter cells are finally formed at the end of the meiosis, which is helping in formation of four daughter cells. So thank you students. We have seen the cell division process in our today's session with meiosis 1 and meiosis 2. Mitosis also we have seen. We'll continue with unit 3 in our next class.